Hey all, so today we're going to be looking at how neurons work. This is C2.2, and we are going to be looking at pages 428 to 446, and then as well as 224 and 225. This is related to sodium potassium pumps. And what we're trying to think about here is how our electrical signal is generated and moved within neurons. So first we have to understand what a neuron looks like, and you can see an image of a neuron here. And so we have the cell body. This is processing information. The dendrites are receiving a signal. The axon sends the signal, and then it sends a signal to the axon terminal, which releases neurotransmitters and are sent to the next neuron. On the surface of the axon are these myelinations. So this myelin sheath created by Schwann cells, which are these fatty deposits. And what's happening here is this myelin sheath allows the speed of the neuron signal to be increased by about 100 times. So we can see here normal speed is 1 meter per second, but a myelinated neuron is going to travel at 100 meters per second. So many layers of this phospholipid prevent depolarization, so that's voltage change, from happening across the whole axon. Instead, the voltage change is only happening at these nodes of Ranvier, which is called saltatory conduction. And so the signal is jumping from one myelin node, one node of Ranvier, to the next. And there's many sodium and potassium channels in the nodes, and we're going to learn what those sodium and potassium channels do on the next slide. So these dendrites are short branched nerve fibers that are going to send signals within the brain or the spinal cord. Axons are long fibers that transmit from the toes to the spinal cord, or from the arms to the spinal cord. And then motor neurons would have the axon terminals connected to muscle fibers. So then that would signal a muscle fiber to contract. So firstly, when we're thinking about neurons, we have the inside of a neuron, and then we have the outside of a neuron. And inside of the neuron, there is a resting potential. So compared to the outside, the fluid that just encases that cell, the cytoplasm is usually negatively charged inside of the neuron compared to the outside fluid. And so there is a potential voltage difference because of a charge difference on the inside and the outside. And there are protein pumps inside the membrane here that are going to use ATP, and they are going to move sodium and potassium in different directions. And they're going to cause an overall charge difference of negative 70 millivolts on either side of the membrane. And so those sodium potassium pumps are pumping sodium out into the fluid surrounding the neuron, and they're pumping potassium ions in. But you can see here 3 sodium go out and 2 come in. And so the concentration of the sodium ions is going to be higher than the concentration of the potassium ions. So there's not as much plus in here as there is out here, which then means there is a charge difference. And that's what a voltage potential difference is. And then additionally, ions leak back through the membrane. All of them leak a little, but potassium leaks back out at about 50 times more than sodium would. And so these potassium ions are leaking out of the cytoplasm, making it even less positive in the cytoplasm. As well, there are negatively charged proteins inside the nerve fiber. So this is more negative compared to the outside fluid. And so we end up with this negative 70 millivolt charge. So in order to send a signal down a neuron, we need an action potential. And an action potential is achieved when a certain amount of signal is sent and it triggers depolarization. And so depolarization and repolarization comprise this action potential. So depolarization, enough signal is sent. Usually this is via neurotransmitters. Enough neurotransmitters have connected to the neuron. And so then the neuron starts to open its sodium channels, and sodium is going to start flowing into this neuron. And that's going to make it about 10 times more concentrated sodium on the inside, and it's going to change the charge. So it's going to suddenly become positive 30 millivolts, meaning there's more positive charge on the inside than on the outside. And then repolarization, so sodium's just flown in, and now the potassium channel's open and sodium flies out. And as a result, it's going to recreate that negative 70 millivolt charge difference, but the sodium and the potassium ions are now in the wrong place. So sodium and potassium need to be returned to their resting locations via pumping using sodium potassium pumps. And that's going to use ATP to reestablish resting potential. Sometimes some organisms have wider neurons, and that allows them to send signals faster. But remember, we haven't learned this yet, but with these signals, it is an all or nothing process. So it has to signal, signal, signal. Once the signal is strong enough, then all the sodium channels will open, sodium rushes in. There's no halfway point going on here. So this is the sodium potassium pump that's going to reestablish resting potentials. 3 sodiums, that should be a small a there, sorry, are pumped out, and 2 potassiums are pumped in. This is the sodium potassium pump here. And one cycle of this uses ATP. So 3 go in, 2 go out using 1 ATP, and this is going to reset the potential. This is called an exchange transporter because it transports things in and out in opposite directions. And this action potential will be triggered if a threshold potential is reached. So this is relating to the previous slide, meaning depolarization and repolarization will be achieved if there's enough initial signal. So when we are looking at the process of transmitting, sorry, just a sec, transmitting a signal down a neuron, the axon is quite long. And so we need to get the sodium channels opening along the stretch of the neuron, of the axon. And so this is called local currents. And what ends up happening is the sodium ions first come in, sodium channels are open, and they're going to start diffusing down the axon. And as they diffuse down the axon, they trigger a threshold potential. And so we're going to see this in the next slide, but the potential difference between the inside and the outside of the neuron is negative 70 millivolts. But in order for them to send a signal, they need to get to negative 50 millivolts. And so here's what's happening. The sodium ions are moving down the axon, and they are reducing the amount of charge difference. And so that triggers the next sodium channels to open. Sodium flows in, and the local current continues where the sodium is diffusing down that way, and then more sodium is diffusing the opposite direction. So it's lessening the charge difference on the outside and increasing the charge difference on the inside. So sodium channels only open at certain points, and these are related to these local currents, and they're called voltage-gated protein channels. So for a depolarization to happen, we must get to that negative 50 millivolts, and then all the sodium gates are going to open. So therefore, the signal is sending an all or nothing. And then there's a separate voltage-gated channel for potassium that's going to open at negative 40 millivolts. And so what's happening are these channels are specific to the atoms that they move, and they're related to the size and bonding capacity of these proteins. And so what's going on here is the sodium ions move through these local currents down the axon on the inside. And once they trigger the sodium channel to open, it opens and sodium flows in. And then this ball moves after about 0.2 milliseconds or 1 millisecond there. This ball is going to move and block any more sodium from moving in. And this is how a potassium voltage-gated channel also works. And potassium is surrounded by water, but it must temporarily bond 2 amino acids in the pore to make their way through. Otherwise, it's too big. So that sodium can't get in through a potassium channel, but the ball is going to hold open the channel for potassium. So this is a sodium channel that we're looking at, but you can see a picture of the potassium channel on page 439 of your textbook. So to summarize, we have sodium potassium pumps establishing a concentration gradient. It's called the resting potential that allows a neuron to send a signal. It's negative 70 millivolts here. And when the threshold stimulus is reached at about negative 55, negative 50 millivolts there, depolarization is going to happen, and then repolarization is happening. And depolarization happens when sodium rushes in, and repolarization happens when potassium rushes out. And then pumping, resetting the resting membrane potential. Sodium and potassium need to be put back in their separate locations. And so when they switch places, we've now gone from a negative 70 millivolt charge up here to a positive 30 charge. And this is when the gated voltage channels are going to open for potassium to then flow out. And then we're going to reset it. And signals only travel one direction in neurons, and that's due to the movement of the local currents. Myelination speeds up signaling, and local currents are also triggering the sodium-potassium channels to open. So that's all for this video. Thank you very much for watching.